Hello, folks. Welcome, welcome. Have that up. All right. So, like Quinn said, I'm really excited about this paper. It is. It's really exciting. So this is the report of fertility in a woman with a predominantly 46XY karyotype in a family with multiple disorders of sexual development. So right off the bat, I'll break down some of what that means. Humans have a number of chromosomes in their cells, and it is 46 total, with the last pair, the 23rd pair, being sex chromosomes. We know these as being X and Y, and in quote-unquote women, typical women, you find an XX chromosome. What this means is that this is a fully fertile woman who gave birth to a healthy baby. She actually got pregnant twice, as you'll see in the paper, without issue. She had regular menstruation. She's a very typical woman, but she presents with 46XY karyotype, which means the classically male chromosomes. Am I blowing your noodle yet? I wanna blow your noodle. So we'll skip the abstract because we're gonna really sink our teeth into this one, and we will start with this little introduction here that says normal sexual differentiation in 46XY individuals relies on a complex cascade of numerous genes, many of which have yet to be identified, and this has 11 individual citations backing up this one sentence. You love to see it. There is a lot of info that in just this sentence. We have a lot of history that there's genes. It's not just the Y chromosome. It is a huge complex network of individual genes that produce proteins that trigger other genes in other cells, that turn on markers to induce cell differentiation in a specific way. It's huge. It's weird and it's complicated. And there's 11 individual citations all saying this. And this is what they start with. Cool. So let's go on. Defects in these genes can cause disorders of sexual development of varying severity. Of course, we all know that there are defects in genetics that produce a phenotype that is faulty. What immediately comes to mind is sickle cell anemia. So this is a single point mutation in a gene that codes for the shape of hemoglobin. And in individuals who are heterozygotic, which means they have a copy that is the typical round disc shape of hemoglobin and a copy that produces the sickle cell anemia, they have conferred resistance to and better diagnostic implications or prognostic implications of malaria. So they have some protection against malaria with these sickle cell hemoglobin, because of course the malaria can't get in. But patients who are homozygotic for the sickle cell anemia gene, so this is again one point mutation in one gene that confers this phenotype, which just means the physical manifestation of this gene. In patients that are homozygotic, which means they have two copies of this gene, they only have this sickle cell hemoglobin, and these sickle cell hemoglobin proteins are way less efficient at transporting oxygen. So that's the part anemia. Anemia means you have low blood count, low blood oxygenation. Your blood doesn't work right. And right here, we see these genes can cause disorders of sexual development of varying severity. The external genitalia and Mullerian structures are typically female in women with complete 46XY gonadal dysgenesis in association with streak gonads bilaterally. Because the gonads are dysgenetic and non-functional, spontaneous pubertal development seldom occurs in these women. What does that mean? So this is hinting at a syndrome that maybe you've heard of if you're fans of Hans Wormhat. This is known as Spire syndrome. And Spire syndrome is when these patients have the karyotype 46XY, so they have the XY chromosomes, but they present as female. The only problem with Spire syndrome is it typically manifests as infertile women. They are usually very thin. Their genitalia is usually typically female presenting, but they are not known to go through puberty normally. They usually have to have hormone treatment because they don't have ovaries at all. And that's what this says. And in this case, the gonads are dysgenetic and non-functional. Spontaneous pubertal development seldom occurs in these women. They don't go through puberty. They don't have functional sexual organs. And successful pregnancy is even more unusual. Unassisted pregnancy is unheard of. So this is the first case that has ever been reported in the world. There have been a few instances of fertility in 46XX, 46XY true hermaphrodites. That's another link. This is why I was reading this and going, oh, this could be a rabbit hole that we just go down for hours. But I'm going to stick with this paper for today. But no reports of fertility in a 46XY woman. So there's none. This is brand new. Pregnancy in Turner syndrome is reported to be possible in about 2% of cases. Turner syndrome is 45X, so there is a missing 23rd pair. Turner syndrome is reported to be possible in 2 cases, although it is rare for unassisted pregnancy to occur in non-mosaic Turner patients possessing only a 45X line. This is citing the paper that, again, could just go down for hours. This is true, quote unquote, true Turner syndrome. In patients with 45X and also 45XX, they have cells that are both in some capacity. And this is called mosaicism in biology, or chimerism, I've also heard. Hi, Dove. I can't type. Sorry. Herein, we report the extraordinary case of a fertile woman. Yes, with normal ovaries and a predominantly 46XY ovarian karyotype, which means her ovarian cells are XY. It's mind-boggling. Her stereotypically as female as you get. These are the organs. These are dost thou bleed. These are XY. It's nutty. Who gave birth to a 46XY female with complete gonadal dysgenesis? This is her successful pregnancy that she brought to term, and this is patient 1 in this study. Also 46XY, but more typical of this genotype. You see the typical phenotypic characteristics. So she didn't have proper gonad formation. Her ovaries were one was completely undeveloped, and sorry, I did not pimp myself in the Discord. I really just wanted to dive into this. I got excited. If someone wants to do it for me, that would be great. The karyotype of this phenotypically normal mother was 46XY in the blood, 80% 46XY, and 20% 45X, which means there's a mosaic of this XY karyotype as well as Turner syndrome, X, mono X, in cultured skin fibroblasts. And here's the kicker, 93% XY, 6% X, and less than 1% XX in the ovary. The family pedigree on the mother's side was notable for the presence of 7 individuals over 4 generations with either sexual ambiguity, infertility, or failure to menstruate, including one individual with documented 45X, that's mono X, and 45XY. 45XY means that a different chromosome is deleted, so maybe we'll go into that. And this apparently had the phenotype of having mixed gonadal dysgenesis, which means the doctors couldn't really determine one way or the other which way the sex organs looked. Was it male or female? We would call this intersex now, probably. Both the mother and the 46XY daughter were screened for mutations in a number of genes known to be involved in mammalian testes determination. In all genes screened below, the open reading frame was found to be normal. This suggests that a mutation in a novel sex determination gene or a gene that predisposes to chromosomal mosaicism may be responsible for the phenotype in this family. What all of that means is that all of the male determining genes that they tested in both of these XY women were completely typical and normal for a phenotypic male. So all of these genes were working. They made the proteins they were supposed to make. They were not mutated in any way that would make them non-functional. They were just switched off. So we'll go down to the methods. Informed written consent was obtained from the subjects, obviously, but it needs to be stated. LHRH stimulation test. This is I have over here. I won't get this word wrong because it's a long one. Luteinizing hormone releasing hormone stimulation testing. This is to distinguish between true hypogonadotropic hypogonadism and constitutional delay in growth and maturation. So that means that they are this marker looks specifically for underperformance of the female gonads, the ovaries. Does it produce the correct hormones in the correct amounts? And this is what this test is looking at. ACTH stimulation is another test of hormone releasing. This one is specific to the adrenal glands. This is the again, just pulling it up, the precise word. Oops. Sorry. And then the last one. Yeah, the last one is the HCG, human chorionic gonadotropin stimulation test. This is an interesting one. So this one is specific to male sexual development. This is a hormone that, when injected in the body, stimulates the production of testosterone in men. Really late in development in women, it will stimulate the production of progesterone in some amount, but not to the same degree. So it's normally for men, and looking at how their organs function in a healthy way. So there were some hormone assays. These are just blood work, normal blood work. And then the karyotypic analysis. So this is karyotypic analyses. This is describing what karyotyping. And what karyotyping is they take your cells, normally in some phase of division, preferably metaphase, when the chromosomes are doubled up and they're outside of the nucleus and they're in a nice area to get at, and they take them apart and they look at them and they pair them up. This was done in the past by hand. Yeah, in situ. It was performed on a paraffin slide of the gonad. So they took cells from the ovaries and used this specific probe, the set probe for the Y, and the chromosome X had a different probe. Pretty typical, but this is just how they determined what the cell makeup was chromosomally. Moving on to the molecular analysis. Chapman divisible. Ooh. Yeah, so I've been reading about some of that where the and just for the VOD's sake, Chapman is saying they encountered a dog with a penis who we thought was a cryptorchid. However, when the doctor opened him up, the visible testicle was actually an ovotesty. So that means that the genitalia were both malformed and kind of both. And we actually have what it might be some of that. Again, there's a family history that I want to get to after we cover the rest of this paper. It's really cool. And it's extensive. So maternity testing was performed using STR kit, and then this is the name of the kit and who made it. And they tested specifically 9 STRs, and then these are the STRs they listed using thermocycling conditions. So they did PCR. Capillary electrophoresis was performed according to manufacturer's instructions. So this is running the gel, and products were analyzed using the AB1 blah 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 blah. This is their software and the instrument that they used. This is what a short tandem repeat is in genetics. Is in your chromosomes there are these short tandem repeats, and that is a repeating sequence of DNA that is short. So it might be like a CCGC, and then you see CCGC, CCGC, and you count how many of those repeats there are. And if they line up closely, then you can say that these people are related. And if they line up to enough of a degree, you can say that there's a paternal or maternal lineage between these two. Normally this is done for paternity testing, but obviously they wanted to be extremely sure and not take the mother's word for it, because obviously she was there. But they ran this test anyway, and these are the genes they used. If you want to learn more about them, these are really well documented STRs. This is a really interesting field. They're also called microsatellites. Yeah. Moving on, the Y chromosomes of the proband, and her two parents were typed using the marker Y chromosome allopolymorphism, YAP, as described. And I read through this paper. It's from the late 90s, and it is just talking about a really cool, highly specific and unchanging marker in the Y chromosome. And the Y chromosome, as we understand, is inherited from father to son, typically. In this case, it was from father to daughter. Cool. But this is just another gene that we can look at to determine where this person's Y chromosome came from. Running this test between the mother and father of and they're both known to be the biological mother and father, I don't think is heard of. I don't think this is what we normally do, but they did it, and the entire open reading frame of the sex determining region SRY gene was amplified and directly sequenced as described. So an open reading frame is, very briefly, the sequence of DNA that is a gene that produces a protein. The open reading frame goes from the start codon to the end codon. So using the PCR primers, PCR is the polymerase chain reaction that amplifies DNA in a very controlled way with thermocycling and a heat-stable polymerase. Getting into the weeds here, I want to breeze through this as fast as possible. They did this PCR on the reading for the ORFs of 7 genes known to be involved in sexual development. Those are these 2 genes, the DMRT1 and DMRT3 genes. They were sequenced using lymphocyte DNA, so this was blood drawn from the patient. These are the conditions for thermocycling. Really, really thorough with outlining their exact protocol. Something we didn't see yesterday. Amplification of the coding region for the B-pesky1 gene was performed using PCR. Again, these are the thermocycler conditions, really specific, and we're outlining all 7 of these sexual development genes. WNT4, DAX1, F2, DAX1. These are all coding for some protein that relates to sexual development, normal sexual development. SF1, SOX9, TISPL. We'll skip through that. Case histories. This is where things get juicy. So the patient 1. This is who came into the hospital first, and she presented from Croatia. Was the product of a 49. This is going way back. Sorry, 39-week gestation, delivered by cesarean section due to paternal hip fracture. So the mother, at some point in pregnancy or before, had broken her hip and couldn't deliver the baby vaginally. So there was a C-section. Totally normal. Birth weight 3.8 kilograms, 52 centimeters in length. Breastfed for a year. Sought medical attention. This is why she came into the clinic now. At 17 years old because of lack of breast development and primary amenorrhea. That means that she wasn't going through puberty. She had not had her period yet, and her breasts were underdeveloped. They go on to say that intelligence was normal, determined by her standing as a top student in her class. I would say that is not just normal intelligence, but above normal intelligence. But they want to be objective. On exam, she was articulate, tall, thin woman. The height was 187 centimeters, totally normal, with a mid-parental target height of 181 centimeters. This is the height that doctors might think she would be at her age based on her parents' heights. Again, totally normal. Her weight was 68 kilograms, thin. She had a normal frontal hairline, so there was no obvious sign of balding. Okay, Magnesar, I'll see you. There was mild facial acne, but no facial hair. She's a teenager. She had Tanner 1 stage breasts and Tanner 4 stage pubic hair. The Tanner stage, as I understand it, is a way of breaking down secondary sex characteristics, and the end stage is stage 5. So stage 4 pubic hair is bordering. It's probably where it should be, quite frankly, at 17. Maybe a little underdeveloped. But Tanner stage 1 breast means almost undeveloped. External female genitalia were normal, without clitoromegaly, which means her clitoris wasn't enlarged, or labial fusion, meaning she had normal labia that were parted. The vaginal introitus was normal, meaning she had a very normal vaginal opening. Pelvic examination revealed a hypoplastic uterus with no palpable gonads. That means her uterus is underdeveloped by the doctor feeling it, and no palpable gonads, meaning they couldn't find her ovaries. by feeling around in there. Bone age was 14 at a chronological age of 17. Again, normal. Bone age tends to lag because there's a turnover. Yeah. Karyotype and peripheral blood performed twice was 46XY. So they did this twice to be sure, and both times she was 46XY. Further investigation revealed that multiple family members on the mother's side had ambiguous genitalia, infertility, or problems with sexual identity. That's figure 2. I have that pulled up in this tab. This led to the decision to karyotype the mother. So, they wanted to karyotype the mother, which completely expected. You know, they want to see where this genetic abnormality came from. So, patient 2, the mother. This 52-year-old woman, this 52 phenotypically normal woman underwent normal pubertal development and reached spontaneous menarche at age 11. She had her first period at 11, and she went through puberty just fine. She had a history of 2 pregnancies, the first of which resulted in a spontaneous miscarriage. That happens, unfortunately. Her second pregnancy was uneventful, except that her daughter was delivered by cesarean section due to a recent hip fracture in the mother from a motor vehicle accident. Again, totally normal so far. She breastfed the daughter for 1 year. She continued to have regular menses until menopause at age 49, after which she received hormone replacement therapy for 2 years. Totally bog standard. Physical examination revealed a feminine appearing woman with normal body habitus. There's a picture of this woman. I can assure you she looks like a 52-year-old woman. The height was 177 centimeters. There was no receding hairline or balding of the scalp, no acne or facial hair. Breasts and pubic hair were Tanner stage 5, although pubic hair was sparse. From having looked at the image, she shaved. Not abnormal, but whatever. They pointed out. The external genitalia were normal, with no clitoromegaly or labial fusion. Again, totally normal vulva. Vaginal introitus was normal. There was no fusing of the labia. Everything was fine, which to be expected. She got pregnant twice. Pelvic examination revealed a uterus in a retroverted position with no adnexal masses. This means that the uterus was tilted back rather than forwards. That can pose some risk in delivery. It might make delivery a little more complicated, and it can also be preceding endometriosis, but she didn't have any of that. She was fine. No adnexal masses means that there were no cysts or other growths that were abnormal on the ovaries, the fallopian tubes, or any of the ducts. The karyotype in peripheral blood was 46XY, and they tested this in 20 cells. So her blood is exclusively XY, and she's a completely typical woman. This would be completely unremarkable. Any physician looking at her would say, you are definitely a biological woman, whatever, but no one would bother to run the karyotype to see if she was XX, because why would you? She's totally normally functioning. But they did, and this is what they found, and it's extraordinary. So, going back to the patient 1, repeat karyotype revealed the following. In the blood, it was 100% 46XY. So every cell that they tested in the blood had this XY chromosome in the daughter. In the skin, it was also 100% XY. And in the gonad, it was 100% XY. This 0.75% could be anything, but it's really statistically almost insignificant. So, hypothalamic pituitary gonadal axis. This is the hormone test that they ran. The LHRH stimulation test was consistent with gonadal failure or absent gonads. So, unfortunately, this means she did not respond to the follicle stimulating hormone, which means she would probably never undergo normal menstruation and ovulation, and probably never have biological children in the normal way. But this is the typical Spire syndrome female presenting person. They usually have, if not no gonads whatsoever, no ovaries whatsoever, they have like a smear of cells or scar tissue, or it's severely underdeveloped to the point of being, yeah, a legit doctor saying biological woman. HCG test. So this is the stimulation test normally used to see testes function. Revealed high baseline testosterone with little response to HCG. Now, in boys that are usually given this HCG stimulation test, what HCG stimulates is the production of testosterone, and so you'd see like a big spike in the level of testosterone over the 2 to 3 days. It's a 4-day long test. Adrenal function, no evidence of steroidogenic defect was demonstrated, so she had normal functioning adrenal glands. Everything wrong with her was isolated to the gonads, to the sexual organs. So this is the pelvic ultrasound. They did an ultrasound wand on the patient's pelvis to look at the organs. So the normal kidneys and a left extrarenal pelvis were noted. This just means that she had normal kidney shape, size, mass, and everything, but that there was a little extra bits on the left kidney. I looked it up on Google and did a lot of searching around. This is totally normal. Extrarenal pelvis. It's totally normal. It's fine. It doesn't usually lead to anything wrong with the kidney function. The uterus was hypoplastic on ultrasound. This means underdeveloped. A small left gonad was present. No gonad was noted on the right. So she had one small, probably insignificant left ovary and nothing on the right side. So there was a surgical pathology done. She underwent laparoscopic gonadectomy, which means they didn't cut her open, but they put a long needle and maneuvered around instruments to remove her ovaries. No gonadal tissue was identified on the right, so there was completely nothing there, although there was a normal fallopian tube on that side. So it appears as though the normal uterus and sexual organs reproduced fine up until making the ovaries. Histology, so this is looking at cell staining and to different markers to see how they respond. On the left, she had a small fragment of fibrous tissue with a small focus of ovarian stroma. Inhibin staining was positive. So this means that the tissue that was there was definitely ovarian tissue, and it responded in the normal way. But that she also had some scar tissue left there. Yeah, this is the daughter. And then her serum testosterone, repeat testosterone after gonadectomy, was within the normal range. On to patient 2. So they did repeat karyotypes and revealed in the blood 100% XY. And in the skin, interestingly, there was some mosaicism noted of 46XY at 80% and Turner syndrome mono X at 20%. And they did this on 50 nuclei. In the gonad, there were 46, 92.9% XY, 45 mono X at 5.9%, of below 1% for XX and below 1% for XXY. They looked at 1,000 nuclei in the gonads. This is because I think it says later on in the paper that the mother also had her ovaries removed because she was already postmenopausal, but it's been shown in the past that when you have ovary cells that are XY, they can become cancerous after menopause, which normally doesn't happen with XY ovaries. So this is the hormone treatment tests. So the LHR stimulation test was consistent with menopause or absent gonads, meaning that the response to this hormone wasn't observed, but that's because she had already gone through menopause and no longer had functioning in that way ovaries. So her gonadal function, this is the HCG test, revealed no evidence of testicular function. She didn't have testes, so totally expected. Her adrenal function were totally normal. They did a pelvic ultrasound on her. So this is the uterus measurement, and it was normal in echo texture, meaning the organ wasn't obviously lumpy and bumpy and incorrect. The endometrial stripe measured 4 millimeters. So in women who are still ovulating and still going through regular menarche or periods, cycle is normal, we'd expect this to be We'd expect this to be in the range of 8 millimeters thick. And in women who are postmenopausal, this can be as small as 2 millimeters thick. They note that 4 millimeters is thickened. The right ovary measured this with normal venous and arterial flow, so it was totally fine. The left ovary was almost the same, with again normal flow. There was no free fluid, meaning the ovaries or everything else wasn't bleeding. Everything is good. MRI of the pelvis. The uterus appeared mildly atrophic and had a mildly thickened endometrial stripe. You would expect the uterus to become atrophic after a while because it's no longer in use. Muscles tend to waste away when they're not being used. There were probable small myomas in the lower uterine segment. This just means some benign growths. Both ovaries were atrophic with no follicles or masses noted. Again, she had already gone through menopause. The music's loud. I'll turn that down. Yeah, so this is totally typical of a woman of her age. Surgical pathology. This is where it says the mother agreed to undergo gonadectomy because of increased risk of cancer in the gonads containing a Y chromosome. The internal structures were those of a normal woman. Right, there's a picture of her gonads from the laparoscopic surgery. No Wolffian remnants were seen. Wolffian remnants, I looked this up, are like leftover pre-maturation tissues, structures relating to normal uterine development. So the fact that none of these were seen means that her sexual reproductive organs were totally fully developed. The uterus and fallopian tubes were left intact, and the ovaries were removed. So this was just a gonadectomy. They did not do a full hysterectomy. Histology. Pathology revealed a histologically unremarkable... Oh, yeah, they do. Yeah. Your muscles will waste away when you don't use them. Pathology revealed a histologically unremarkable right ovary with several corpora albicans. This means whitening of the flesh, suggestive of previous ovulation. This is just what is expected to happen. When the tissue is left there, it tends to get attacked by the immune system, white blood cells, and that turns it white. Totally normal. The left ovary contained fibromuscular tissue with ovarian Hiller cells, and an immunohistochemical stain for inhibin showed a focus of positive cells confirming the presence of ovarian stromal elements. So this means that the ovary had functioned in the past completely normally, but now was not because, again, she had gone through menopause. Repeating the same thing, beating the same drum. Family history. And I think I might go right to figure 2 after this. So the family history. There's a remarkable family history of ambiguous genitalia and infertility affecting both phenotypic men and women across 4 generations in the mother's family. The daughter inherited her Y chromosome from her father, which is pretty cool, thereby excluding involvement of the Y chromosome in the development of sex reversal in this family. So this is saying, what if the mother's Y chromosome had something special that allowed her to reach full sexual development, but it wasn't given to the daughter who inherited the Y chromosome from her father? There might be something there. The pedigree is strongly suggestive of X-linked inheritance of this phenotype, although autosomal dominant sex-limited transmission cannot be excluded. So X-linked inheritance just means that it's passed on your mother's side, and autosomal dominant means that when 2 copies of the same gene are in the same person, that the dominant one will be the one that's used. An autosomal dominant trait is eye color. If you have melanin in your eyes, it's brown, and that trait is autosomal dominant. Just so you know how that works. Sex limited means it only presents in one sex or the other. It's super tricky, because this paper is like overturning some of the definitions we know about sex. But there you go. Again, they're covering their bases here. These authors, they're being a little careful. They're saying it could be this, could be that. And I want to go right to the figure 2 here, and I'll pull up this. And I'll have to switch over to the display so we can see that. This is the family tree. These arrows are pointing to the patients, but we'll go through. I'll point them out as we read them. Family members. Family members 2, 3. So that is generation 2, number 3. And as we can see from the bottom here, these grayed out squares and circles are the affected phenotypic men and women. Circle is female, square is male, and X through the part in the tree means that this person is deceased. So, Gen 2, person 3, is a woman with a masculine appearance, no breasts, infertile, moved away from hometown because of unacceptable appearance, and died during World War II at age of 62 years. Generation 2-4 is this person's sister, who died at age 76. Apparently totally normal. She went on to have 2 kids who were affected by this disease, or this genetic condition, at least. Right, 2-6, woman who died at 68 years old, unaffected. 2-7 was the man she married, who died at age 64. They went on to have children who were affected, including the grandparents of the original patient. Yeah. I will warn you right at the front, right here. Well, not at the front, but before we get too much deeper into this, there is mention of suicide, and as a product of its time, if you were not completely the typical man or woman, you weren't accepted yet, and we see that in this history. So just fair warning off the top. 3, 5, and 6. So this is generation 3, patients 5 and 6 on this list. So these were born to unaffected phenotypic. A sister of a person who was affected and died, but did not present with the condition. She gave birth to 2 daughters who had the condition. And these two are reported to have hirsutism, which means they were overly hairy. You know what that means? Uh, they might have had a mustache. You know, terrible. 3-9. So this is in generation 3, person 9. This is a daughter of this pair, which were very prolific. Presented with the phenotype 3-9. Yeah, ambiguous genitalia with hirsutism, a beard, was raised as female. The family was ashamed of her and hid her from public. Died at age 55. Tragic. You know, this is a person we would call today intersex. I'm quite confident in saying that much. 310 was a man with confused gender identity. Here's the trigger warning. Who was infertile and committed suicide at age 24. This is the phenotypic abnormality rearing its head. So 313 is a normal man who died at age 70 years old. Obviously, after this was taken, otherwise he'd be X'd off. 314 was a normal woman. So we go to generation 4. 3. This is the dad of the original patient. 46XY, fertile man. Typical man, apparently. 4-4 was a fertile woman with predominantly XY ovary. This is patient 2. This is the person we're interested in, this overall history. 4-5. So this is patient 2's sister. Sorry, I lost my place. Yeah. 4-5, normal XX woman. 4-7, this was a woman with an absent uterus and ovaries. This was established outside the facility and outside the city, and the history was obtained from patient 1. She died before this was taken. Let's see. Patient 1. She was on hormone replacement and died at age 42 from multiple sclerosis. 4-8, normal woman. 4-9, the normal woman's husband, completely typical. 10, fertile woman with XX chromosomes. 4-11, normal XY man. 12, XY man. 27, way out here, is typical XX fertile woman. Generation 5 now. 5-3. This is patient 1. This is the woman who is the teenage girl who was XY with complete gonadal dysgenesis. 5-5 is a normal fertile woman, XX. 5-6 is an affected man. So what happened here? We have ambiguous genitalia, intersex apparently, bifid scrotum, which means there's a cleft in the scrotum. It doesn't descend fully all the way. And hypospadias, hypoplastic testes and scrotum, stretched penile length of 5 centimeters. This means all of the genitalia is pretty underdeveloped. So this is that phenotype of having either ambiguous genitalia or underdeveloped genitalia. There are high gonadotropin levels in the blood. So this man is producing a within normal range follicle stimulating hormone, which is specific to ovulation in women. Pretty interesting there. Obviously infertile. Testosterone looks low, but there's also a blood estradiol and prolactin levels, which are strange and not typical. Here we go to 5'7", a normal woman, 5'8", a normal man, 5'9", normal woman. They have 2 kids between them. 5'10", normal man. 5'11" is XX, normal woman with normal hormones. 12, normal woman. What else? 16 is a normal woman. 20, normal man. 21 is an infertile woman, interestingly. Tried in vitro fertilization without success, they note. 23, normal male. 528. So this is another individual with the affected phenotype. And what do they have? They are 46XY, 45X. So this is a mix of the Spire and Turner genotypes. Mixed gonadal dysgenesis, gonadoblastoma. So this is what the patient 2, the mother, was trying to avoid. This woman obviously had XY chromosomes in her ovaries. What ovaries there were. It says mixed gonadal dysgenesis, which means maybe one was developed and sort of functioning, and the other was probably not there at all, and likely developed into a tumor. So we're going to generation 5 now. This is the patient 1's generation. 5'1" is a normal woman. 5'1", we're going to generation 6 now. Sorry, can't read. Roman numerals. So this is the generation after the original patients. This is the youngest generation. We don't have any observed phenotype in these children yet. These were all personal examinations done by one of the authors of this paper. Hi, Liza. So, the genetic evaluation. Maternity was established at a probability exceeding 99.15%. They couldn't just take the mother's word for it. They had to do genetic analysis. The analysis of the Y chromosome polymorphism YAP revealed that this insertion was present in the Y chromosome of the mother, defining her Y chromosome haplogroup as D/E, and the insertion was not present in the Y chromosome of the daughter or her father. This means that the Y chromosome came from her father, and the Y chromosome in her mother was completely different and had this unique insertion. Sex do be funky. These data indicate that the daughter inherited the Y chromosome from her father, as I said, and the sequence was identical to that of a normal male. The molecular analysis of the coding sequences of 9 genes known to be involved in sexual development, and there are those genes, revealed coding sequences in both the mother and daughter that are identical to normal reference sequences. This means that in both of their Y chromosomes, in these phenotypic women with at least there's development insofar as externally you were unmistakably female. But they had Y chromosomes that were completely normal and in a male would make them masculine. They would produce the secondary sex characteristics of a male. They'd have testes that worked. They would have proper hormone levels. They'd go through puberty with the beard and the ass hair, all that nonsense. But they're women. So what's going on there? Don't know. The authors looked for polymorphisms and didn't find any, meaning there were no obvious breaks or switches in the normal gene and the normal gene sequence. These were almost textbook, apparently. So we go on to the discussion. Although there have been reports of fertility in 46XX and 46XY true hermaphrodites with ovotestes, This is another paper that I might do at a later date. And in patients with mosaic and non-mosaic Turner syndrome, which again is another paper that I'm interested in reading, because Turner syndrome is like, it's one of those classic examples brought up in biology class when talking about chromosomes and sex chromosomes specifically. It's the mono X one. Yeah. You're in for science. So we believe this to be the first report of fertility in a woman with a predominantly 46XY karyotype in the ovary. The fact that this mother had normal functioning ovaries, menstruated regularly, and achieved unassisted pregnancy twice is, as they say, remarkable. Additionally, her hormonal findings are hormonal findings are compatible with a normal menopausal woman. Of course, it should be noted that the incidence of normal fertile females who have this is a good sentence of normal fertile females who have an X46XY karyotype is not known because it's not routine to check the karyotype in fertile women. Of course, you're not going to look at this. Why would you? Everything is functioning as it should. Why would you go and look? So we don't know. This could be a huge population of women out there with this 46XY karyotype that we just don't know about because we're never going to test them. And this is a big takeaway from this paper, is that we don't know that much. Obviously, if you are to be someone who constructs this weird binary, this one paper, this one family history alone is enough to shatter that, because if it's a true binary, then there wouldn't be an outlier. That's not how binaries work. You're either or, you're not also. So moving on, although the demonstration of 5.9% of mono X cells in the ovary is difficult to interpret, most cytogeneticists agree that 5% does not indicate mosaicism, meaning that the level apparently isn't enough to qualify as you have two distinct karyotypes present and they are overlapping to some degree. But the finding of 20% 45X cells in fibroblasts culture from the skin indicates that she is mosaic in the skin. So the cutoff is somewhere between 5 and 20%, apparently, or higher, Individuals with a karyotype of 46XY and 45X, so this is normal male and mono X, usually have ambiguous genitalia or a male phenotype, although occasionally they can have the Turner female phenotype. The Turner female phenotype is small, usually thin, and I believe, I didn't look this up, but I was pretty confident. The really hallmark example of Turner syndrome in women is that they have webbed skin or between the neck and shoulders. Anyway, moving on. Our case is unique, however, because the presence of bilateral ovaries, meaning two, and they're symmetrical with each other, or unassisted pregnancy has not previously been reported in this form of mosaicism. So this is totally new to science. Moreover, ovarian cells were predominantly 46XY. The small percentage of X, 5.9% out of 1,000 cells counted in the gonad, might be due to artifact or technical error. So they're saying maybe it isn't even, 5 or 6% of the cells are mono X. It could just be that we messed up there. Could just be 100% XY. So pregnancy is believed to occur in about 2% of women with Turner syndrome. Another paper that would be fascinating to read says, although fertility did occur in a woman with mosaicism of an isodicentric Y chromosome. This is pretty cool. Isodicentric in this context means that the Y chromosome, when it's split during normal cell division, it forms kind of a bundle in the middle that is normally split in half equally between the two when the cell divides. However, it didn't. So each copy that's produced in the divided cells has this kind of remnant shield in the middle, and it results in the chromosome itself being awkwardly bent and presumably, at least in this case, inactivated to at least some extent. We believe that our case of fertility in a female with a predominantly XY karyotype in the ovary is unprecedented. Of note, XY female wood lemmings, so this is a completely different species, carrying an XP mutation, are fertile and produce X-containing oocytes. So this is just a side note that this happens in nature, but not to a known degree in humans. So what the authors are playing at here is that maybe there is an X mutation in the mother that has something that allows her to have this XY karyotype and produces typically XX phenotype. There have also been reports of potential fertility in XY sex reversed mice. This is another probably paper that I would read on my own time. I don't know how good it would be to stream. So in the B6, this is a region of the mice chromosome. Y-DOM, sex-reversed female mouse, almost all of the XY female mice, although they lack estrous cyclicity, which means they don't go through heat, are able to mate and ovulate after treatment with gonadotropins. So meaning when the researchers treated these mice with normal hormones, they were able to go through normal reproduction cycles. Pretty cool. Likewise, fertility has been described in XY female horses. So this is just, they're piling on here. This happens in nature. We know it happens in nature. This is the first time we've seen it in humans. And we like to separate ourselves from nature when really, are we? I don't think so. We're monkeys. Or, apes. So the fact that this mother gave birth to a 46XY female is even more remarkable. Because, what are the chances, It's crazy. However, the daughter's clinical picture, unlike that of her mother, is more typical of a 46XY complete gonadal dysgenesis, meaning that she is underdeveloped for her age. So it goes on, in which spontaneous puberty is rare and fertility is unreported. So this is what you'd expect with a 46XY, quotes, fire syndrome female. The significant family history of ambiguous genitalia and sex reversal across several generations presents a unique opportunity to explore the genetics of sexual differentiation and perhaps identify a novel gene involved in gonadal determination. Of course, this family is like the first in the world to have this kind of, it is an abnormality. It is different from the norm, but it's not unheard of in nature. So yeah, obviously the authors are right in saying that. Yeah, there could be a novel gene involved in gonadal determination, and it's somewhere in them hills. Genetics is crazy new, and there are breakthroughs every day. The mother and daughter were both screened for mutations in a number of genes that are either known to be necessary or excellent candidates, or excellent candidate genes for males in human testicular development. This just means that these genes are normal for typical development of the testes, but obviously they didn't have them. So some of the genes known to be involved in gonadal determination are outlined in Figure 4. So and then there are citations for that. Right, and in Figure 4, they outline that these genes look normal to reference sequences, and would otherwise be functioning fine in men, but not in these two women. Both the mother and daughter had the normal SRY, sex determining region of the Y chromosomes. Um, I've gone through this paper. It's older. It's from the 90s, and it identifies this SRY gene as being probably the big factor in determining placental, not placental, embryonic development, sex differentiation. So like this is like a switch on the Y chromosome that makes everything go masculine. And the fact that the mother and daughter had this sequence and it was their complete working fine is really it's unusual to the greatest degree that I can state. It is strange, it is cool, it is weird. So moving on, the daughter inherited the Y chromosome from her father. Obviously, this would be like a 1-to-1 transfer. This is a Y chromosome in her dad that works, has made a sexually productive male, and it was just given to her 1-to-1 to make a female with underdeveloped ovaries. Just wild to me, thereby excluding involvement of the non-recombining male-specific portion of the Y chromosome, including the SRY gene, in development of sex reversal in this family. So there was no obvious sign of weirdness in the meiosis. There's a lot of cool crossover events in not like movie Marvel crossovers, but genetic crossovers where as the genes are pulled apart twice without being copied, there's some of the chromatids, the arms of these structures cross over, and where they cross over splits, and you'll have some part of the chromosome from dad and some part of the chromosome from mom left into the gamete that eventually becomes you. And this is what leads to genetic variation and evolution. So included in the screen were these normal genes. When deleted, these genes are associated with male to female sex reversal. So when these genes aren't there, we've seen in probably mice studies or maybe even human studies, I don't know, I haven't read the papers yet, that there's male to female sex reversal. So this would be if you had a phenotypic woman presenting with an XY karyotype, you would look immediately for these genes. Are they deleted? They were not deleted in these two women. The analysis of another gene that is associated with gonadal dysgenesis, it's this one, this TISPL gene, also revealed a normal sequence. So if this gene is funky in any way, then the genitalia doesn't develop normally, but this is not to blame. So mutations in this gene are also associated with sudden infant death and testicular dysgenesis in an Amish family. Amish have funky, weird genetics. They have a very small population base. You tend to see these kinds of mutations happen in populations like that. So this gene, the BPESC1 gene, which exhibits testes-specific expression and is located within the homologous region in the human at 3q23. So this is chromosome 3 on the Q arm at the locus 23. Was normal in both mother and daughter. So they're saying that this is a gene that has testes-specific expression, meaning if it was in a woman, it wouldn't have any function because there's no testes, but it's normal in both mother and daughter. So this gene is there and should be expressing to produce testes, but isn't. Moving on, the DAX1 gene is located on the XP22. I've never seen X written that way, but okay. Maybe they don't know a specific chromosome it's on. Or no, it's the X chromosome, of course, and appears to be necessary for correct testes determination, and in the mouse at least, necessary for the upregulation of the SOX9 expression. So this means if this gene is there and correct, then you would expect to find, along with other markers, of course, that testes would develop in utero. And again, this gene also upregulates another gene expressed that they looked at. I'm not sure what the SOX9 gene is. I assume it has something to do with sexual development. The gene WNT4, we're getting in the weeds again, I'm sorry, is critical for known ovarian and female sexual development. A mutation in WNT4 leads to Müllerian duct regression and virilization in a 46XX female. Right, whereas duplication of the locus leads to sex reversal. So a mutation leads to abnormalities, and a duplication of the gene leads to sex reversal. The mother and daughter had normal genes without any mutations in this gene, so they had presumably the right cards in the deck to make normal functioning ovaries, especially in the mother, but it wasn't the case with the daughter. But it also didn't. It wasn't duplicated, so he can't say, "Oh yeah, that's why she is X, Y, and female." So in the XY mouse, the absence of WNT4. Oh, do share, Liza, if you have an interesting sex-based factoid. The absence of WNT4 is associated with Sertoli cell differentiation, suggesting that the gene is involved in mammalian testes determination. So the Sertoli cell differentiation has to do with production of testes in, again, development as a fetus. Sequence analysis of both DAX1 and WNT4 genes revealed normal wild-type male coding sequences in both the mother and daughter. Wild. The desert hedgehog gene, DHH, is a member of the family of signaling genes, playing an important role in morphogenesis, blah, blah, blah. Mutations in this gene are reported with partial gonad dysgenesis and in women with complete gonadal dysgenesis. So this is the author stating that this DHH is maybe a candidate, at least in other cases. Other possible genes to explore in this remarkable family are the FST and bone morphogenic protein genes, which are both expressed in mouse embryonic ovaries and appear to be important for ovary making or the organogenesis. So FST acts after WNT4 to inhibit the formation of XY specific solemic vessel and to maintain germ cell survival in the cortical domain of the ovary. All of this is to say that these genes are important in both making the ovaries from cells, period, and ensuring that the ovum survive and are healthy. So as I say, there's a Russian town that 1 in 72 men are born effectively AFAB, but are male and eventually develop male genitalia as part of puberty. Interesting. Yeah, so there's a bit of that in this case history, as was stated in the case history section. There were ambiguous genitalia at several points along the family tree, and one surviving member, or the case history of a member that survived, reported bifid scrotum development and I think it said hypoplasia of the testes, meaning that the cleft in the scrotum is there that would eventually become the vaginal opening if left to develop that way, again, in fetal development, but not as a mature adult or a mature baby. If I can say mature baby in the same sentence. So again, the authors point out that mutations in these two genes can also lead to sex reversal in mice, but they weren't. They are potential candidates to be explored, but they weren't here. Alternatively, because at least one member of this extended family, at least one member of the extended family has 46XY 45X mixed gonadal dysgenesis, right? Disorders of sexual development in this family may be due to a mutant gene that predisposes to chromosomal mosaicism and mixed gonadal dysgenesis. So there's a gene somewhere in the family that might make them have two different and slightly different karyotypes that are present at the same time, but in different amounts, that may be the cause of the underdevelopment of their sexual organs, or the absence of development of sexual organs, as in the case of patient 1. So in either scenario, the transmission of the phenotype is strongly suggestive of X-linked inheritance. Again, although autosomal dominant sex-limited mutation can't be formally excluded. So they still aren't sure either way, but it appears that there's something on the X chromosome. Various studies of different XY gonadal dysgenesis have suggested the existence of an X chromosome locus that is necessary for testes determination. So these are a couple more papers that are saying there's a gene on the X chromosome somewhere that determines whether testes are made or not. Perhaps on proximal XP. So the P arm is the long arm of the X gene. The latter finding is consistent with observations of deletions of XP associated with 46XX SRY negative true hermaphroditism. What does that mean? That means that someone is presenting with XX and somehow has both the gonad developments of male and female. I think that's what Faye said she has, something like that. So this is more guesswork by the authors. Look at these places. Maybe there's more to find there. We're almost at the end here. This last thing is the serendipitous discovery of a predominantly 46XY karyotype in this fertile mother of a 46XY daughter suggests that perhaps all mothers of 46XY SRY positive females with complete gonadal dysgenesis should be carefully examined for an XY karyotype as well. So this is saying that we should look at karyotypes more. And they go on to say this extraordinary family affords an exceptional opportunity to investigate potential factors that can induce ovarian differentiation and function without the XQ critical region. So this is saying that this family has something cool and different about their X chromosome that we haven't seen or studied yet, but has led to the full development of normal ovaries. Meanwhile, DNA has been obtained on 19 family members. 3 were affected and 16 were unaffected across 3 generations in hopes of identifying the etiology of sex reversal in this interesting family. So they're still figuring out what is causing it. Linkage analysis is a potential next step as efforts are being made to obtain DNA from more affected family members. Linkage analysis is linking genes to proteins, genotype to phenotype. A genome-wide search for deletions or duplications in this mother and her daughter may serve to uncover novel gene mutations responsible for sex reversal, or may even reveal a genetic cause for the chromosomal mosaicism and mixed gonadal dysgenesis. So this is saying they want to look at the entire genome. Maybe it isn't on the X chromosome. Maybe it's on chromosome fucking 4. They don't know. But the possibility is there. They want to look at everything that they have, and I don't blame them. This is really cool. There's some acknowledgements here, and a whole bevy of references. Now, I don't know if anyone has any questions for me, but that's the end of this paper. That's the end of my stream.